Ok, Eli, si gustas comenzar.
¿Sabes si llegó la doctora Diana Alejandra González?
Eh, no. ¿Listo?
¿No llegó?
No.
Ok. Muy buenos días. La Universidad Autónoma de Baja California, a través de la Facultad de Medicina y Psicología, les da una cordial bienvenida al evento La Investigación Científica como Lenguaje Universal, dentro del programa de internalización en casa, modalidad virtual 2020. Antes de continuar, agradezco la presencia de las autoridades que nos acompañan. El doctor Carlos Vera Hernández, subdirector de la Facultad de Medicina y Psicología, en representación de la doctora Yadira Islas Limón, directora de la facultad. La doctora Gabriela Saldaña Ojeda, coordinadora de la carrera de Medicina. Como organizadora del evento, la doctora Sandra Olvera Hernández. Como invitada especial, nuestra primer ponente del programa, la doctora Liisa Galea, de la Universidad de Columbia Británica. Cedo la palabra al doctor Carlos Vera Hernández para que inaugure el evento.
¿Qué tal? Buenos días. Gracias a todos que se enlazaron para este evento científico tan importante. Yo creo que la ciencia es algo que necesitamos urgentemente en nuestro mundo porque vemos la necesidad de tener análisis objetivos basados en evidencias para tomar decisiones. Y creo que el que tengamos cada día más acceso a conferencistas de talla internacional que nos ayuden a entender cómo funciona el mundo a diferentes niveles es fundamental, ¿no? Y más para un estudiante de medicina que está al frente de esto, un estudiante de nutrición, un estudiante de psicología, que tienen tareas tan importantes como decidir el tratamiento médico, nutricional, psicológico de una persona, ¿no? Entonces me da mucho gusto tener aquí a Liisa Galea con nosotros. Espero que tengamos una interesante plática sobre asuntos fundamentales como el estrés y control hormonal, que creemos que es algo fundamental en la respuesta humana a la emergencia y a las cosas crónicas. Entonces, gracias Liisa por aceptar estar con nosotros y gracias a todos por estar atentos a este evento. Y a partir de esta hora, que es la 9:27, y en representación de nuestra directora, la doctora Julieta Yadira Islas Limón, declaro formalmente inaugurada esta sesión.
Muchísimas gracias, doctor Carlos Vera. A continuación, cedo la palabra a la doctora Sandra Olvera Hernández para que presente a nuestra primer ponente.
Muchas gracias a todos ustedes por haber asistido a este primer día de actividades dentro del programa que se llama la Investigación Científica como Lenguaje Universal. Quiero agradecer el apoyo proporcionado por la Facultad de Medicina y Psicología, así como al programa de internación en casa en modalidad virtual 2020 para la realización de esta serie de actividades durante a lo largo de toda esta semana. In English, welcome at the first day of activities of this area of conference, the Scientific Research as Universal Language. I appreciate all the support for the School of Medicine and Psychology and the program de Internalización en Casa 2020. For me, it's an honor to introduce the first speaker, is Professor Galea. The Professor Galea works in the Department of Psychology and the Center for Brain and Health, director of the graduate program in Neuroscience, leads the Women Health Research Cluster and Scientific Advisor of Women Health Research Institute at the University of British Columbia. Her research goal is to improve brain health for women and men by examining the influence of sex and sex hormones on normal and diseased brain states, such as depression and Alzheimer disease. She was the first researcher for a while to develop preclinical models of postpartum depression and was of the first in to study the long-term effects of motor alcohol in the brain. Dr. Galea obtained her PhD in neuroscience for Western University and was a postdoctoral fellow at the Rockefeller University. She has a lot of distinguished fellowship awards, so I be sure that you can enjoy this talk, and I only say thank you very much, Liisa, for to accept to share your research and your notes in the brain for us. No?
Well, thank you for the invitation. I better share my screen or I can't really do very much, right? So, oh, there we go. Thank you again for the invitation. I wish I could be there with you to enjoy some micheladas. I got introduced to those a few years ago, and I think they're one of the best drinks in the world. So, but, you know, unfortunately, this will have to do for, you know, there's some good vaccines on the horizon. So hopefully I'll be able to come to Mexico sometime in the near future. So for the next 45 minutes or so, I want to talk to you about some of the research we've been doing on stress and sex differences on the effects of depression and treatment. But before I do, I'll just very quickly define what I mean by sex versus gender. So when I'm talking about sex, I'm talking about the male or female biological and physiological characteristics that differ by males and females. And gender is more of a psychosocial construct, which I'm showing a picture of my Canadian husband showing what an appropriate role is in the Canadian society in terms of housekeeping. This is a bit of a joke. In Canada, probably like much in the world, it's women that are supposed to be doing the housework. But gender is a psychosocial construct, so it's what a given society deems appropriate for men versus women in terms of their role or gender identity. There are a lot of sex differences, though, in the prevalence of brain disease, and some of those are depicted there. And obviously, I'll be talking about depression today. But why study sex differences in disease? I think for many reasons. One is it gives us clues on how the disease develops, the manifestation of that disease, and the treatment. And that last part there, that treatment, is very rarely studied. And I'm going to talk a little bit more about that today. But it also allows us to build better models of the disease. And better models will result in better treatments. And when I'm talking about models, I'm talking about both animal models and also in human disease as well. And if that doesn't convince you, a lot of federal funding agencies these days are asking us to analyze with sex or sex and gender. And I'm sorry, I apologize. I think it's contacts or something in Mexico, and I don't know if they have a sex and gender lens yet, but I assure you they will in a couple of years if they haven't already. The other thing is there was more of an interest in the popular media when the Food and Drug Administration in the US, for the first time ever, indicated that a different dose needed to be used for the sleep drug Ambien. And there was a very good 60 Minutes episode based on exactly this. And I think dose is only one part of it. I think that it really needs to sometimes be a different treatment and different therapeutic altogether. Dose is very obvious. And if you think about it, when you go to the drugstore, your pill bottles don't say take 2 pills if you're a man and 1 pill if you're a woman. But typically men, and I'm showing a picture of my children and their and their lifelong nanny there, who doesn't work with us anymore, unfortunately, but to illustrate a point, which is that men are typically heavier and taller than women are. And so by definition, that should suggest that the dose would be different. But anytime you see a sex difference, that should cue you to think that genetics or hormones at least would be involved. And there are a number of genetic similarities between men. The genome between 2 men is shared, 99.9% of the genome is shared between 2 men. But you can see that 0.1% can make a huge difference in terms of the phenotype of the individual. And the genetic similarity between a man and a woman is even greater. It's 15 times greater a difference between a man and a woman than between 2 women or 2 men. And our XX and XY is represented in all of our cells. It's not just in the reproductive tract, but everywhere in the body and brain. And so it obviously plays a large influence, and one that we don't always consider. Again, anytime you think you see a sex difference, that should cue you to think that sex hormones are going to be involved in some way. And so I have an excuse to put my children back up on the screen. And so I know we're all on the same page. I'm talking about ovarian hormones in women like estrogens, and testicular hormones like testosterone or androgens in men. And obviously it's much more complicated than that. We each have each other's hormones, just in different concentrations. And they work on different receptors that are located throughout the body and throughout the brain. And testosterone is actually even more complicated because it can be converted to the most powerful of the estrogens called estradiol. So it can work on either estrogen receptors or on androgen receptors. Testosterone can also get converted to a very powerful androgen called dihydrotestosterone. And anytime, like I said, sex hormones can affect symptomology and as well as treatment. And I'm giving you an example from the schizophrenia literature that just shows as estradiol levels decline across the menstrual cycle, you see psychotic symptoms in women with schizophrenia go up. And I'll show you some other examples of this. Now, really, I've been studying sex differences, but also the female brain. And why do I think the female brain would be different? I think in many ways it's very obvious. Females have a different physiology due to the fact that they're the ones getting pregnant, giving birth, breastfeeding, providing parental care in most mammalian species. That should suggest, among other things, that the brains are going to be organized differently. And this comes from my late master's supervisor, who showed that the incidence of aphasia was very different between men and women. So men are more likely to get Wernicke's aphasia, which is the inability to comprehend speech, and women are more likely to get Broca's aphasia, which is the inability to produce speech. And that suggests, among other things, that the brains are organized differently. As I've already shown you or indicated, sex differences do exist in a lot of brain disease. Women are more likely to present with depression and Alzheimer's disease. And while longevity does play a role in Alzheimer's disease, it's not the only factor. And men, on the other hand, are more likely to present with Parkinson's disease and as boys, autism. So let's look at the evidence of women being more likely to become depressed than men. So I like this particular graph because it shows the female to male ratio. So anything above 1, females more likely, anything above 2, more than twice as likely. And it also shows across the lifespan. So where it starts to come above 2 with the greatest susceptibility to depression, you can see are the sort of the time of very high ovarian hormone production, suggesting that the unique physiology of females results in these specific periods of susceptibility to depression. It also lends itself to 2 alternative biological explanations for sex differences in depression. One being that women are more susceptible to depression, possibly due to hormonal fluctuations, and men are more resistant to depression. Because I'm an endocrinologist, I'm always going to think it has something to do with hormones. And we've created a number of animal models based on this presumption, and I'll explain some of this as I go along. And the other question we've been very interested in is, do we see changes in antidepressant efficacy by sex, but also by hormonal status in animal models of depression? So not just alone, but in a background of depression or depressive-like endophenotypes. Now, I often get asked this question: do men and women just show depression differently, and that's why you see more women with depression? So to be diagnosed with depression, you have to have 2 of the blue symptoms there, and then 5 out of the other 7 symptoms. And I think somebody with the best last name ever, Angst, did this. If you're studying depression, did this study obviously quite a while ago now, but you can't argue with the sample size. This is based on a number of interviews in 5 different European countries. And they went door to door. So it wasn't just people that went to a doctor's office. And what you can see is once you reach 5 or more symptoms, that's when the female to male ratio shifts, so that women are endorsing more of the sort of typical depressive-like symptoms based on the DSM-5 criteria. Now, there have been a number of notable studies recently looking at sex differences in major depressive disorder related transcriptional signatures in the brain. And what's fascinating about both of these studies that came out is that there's very little overlap. So it does look like there are some very distinct signatures of depression in men and versus women's brains. Now, I've told you that when you see a sex difference, that should cue you to think that sex hormones are involved. And I've already shown you some evidence for that. When you see larger fluctuations in ovarian hormone levels, that's when you see greater susceptibility to depression, so during perimenopause and postpartum. And in men, testosterone is more common with if they have lower levels of testosterone, and that's true in both younger and older men. Now, stress hormones are also involved in depression. In fact, that's the sort of main hormone that would be involved, I'd say. Depressed patients themselves will say that stress was a preceding factor before their depression. And depressed patients have increased levels of cortisol, which is the main stress hormone, and that's been shown in a meta-analysis. But they also have altered diurnal patterns of cortisol release. So this is controls here, and you can see there's a bimodal increase in cortisol release in the morning and then another one around sort of dinner time. But depressed patients have a much more erratic diurnal presentation of cortisol release. Another problem with the stress hormone axis is that the depressed patients show a negative feedback, impairments in negative feedback inhibition. And the way that you can test this is by giving a synthetic glucocorticoid called dexamethasone, which in non-depressed individuals will block the secretion of cortisol because there's this negative feedback system. But in depressed patients, there's an impairment in this negative feedback system such that you see much higher breakthrough, much higher levels of non-suppression, particularly in the afternoon. So suggesting that there's impairments at those different levels of HPA axis. Now, I'll be talking mostly about animal models of depression, and most of the ones that are out there are based on stress and stress hormones. And I think we can all agree it's really hard to ask animals if they have thoughts of death and suicide. Obviously, we can't do that, but we can very easily look at some of the other symptoms of depression. So we sort of call them depressive-like endophenotypes. And because they're animal models, we can also look very closely at their brains to look and to see if we see the signatures or hallmarks of depression. And most of the animal models that are out there are based either on stress, manipulating stress, or sex hormones. So what are some other hallmarks of depression? I've talked about the symptoms of depression, but also the hippocampus appears to be involved. There's a lot of meta-analysis indicating that there's a decreased volume. A stress system, as I've already told you, increased levels of cortisol, but the immune system and tryptophan and the serotonin signaling is also affected. So there's a lot of pro-inflammation in the immune system and increased tryptophan metabolism, indicating lower levels of serotonin. So in this talk, I'm going to try to present to you a sort of a unified theory of depression. As I've already suggested, stress can precipitate depression. We know that biological stress can affect the incidence of depression, prevalence of depression, but it can also impact stress and stress reactivity. And so there must be some, or there might be some modulation of biological stress, sex, to influence stress to depression. Depression, we know, also increases neuroinflammation and decreases neuroplasticity. I'll show you some evidence for that. And antidepressants work to reverse these. But what is less studied is how does biological sex and hormones influence both neuroinflammation, neuroplasticity, the effects of antidepressants on these characteristics. So I'll be talking about that today. Obviously, there are genetic components to depression as well as neurotransmitters and their involvement. I'll talk a little bit about serotonin today. Oh, right, I forgot. I Googled depressed women and I didn't like any of the images, so I chose one from my own photo album, and this is my daughter a long, long time ago now, and she looks, I think, depressed, but she is not. I just wanted to assure you that she's not depressed. It's just a Halloween and her brother wouldn't share candy with her, so that's why she was looking so sad. But I just want you to ensure you that no children were harmed in the filming, that early in the day she was quite happy. So what about the neurobiology depression? Unfortunately, I am very much obsessed with the hippocampus, but obviously it's more than the hippocampus. However, there's some very nice work that's been done in the hippocampus. It has a lot of plasticity, which I'll talk about in a second. But depression itself is associated with a small hippocampus. That's been shown in meta-analyses. It's also related to the duration of illness and not age itself specifically. But as I mentioned, there are many other areas that are involved in depression, notably the regions of the frontal cortex, as well as the nucleus accumbens and amygdala. But in terms of the hippocampus, because it's so plastic, it's a nice area to look at. And if you're thinking about a reduction in the volume, that could be done due to the number of cells in the hippocampus and or neuropil. And it turns out that each of these factors could be involved. So here's a coronal section of a rodent hippocampus. And we see evidence that both sex and steroid and stress hormones can influence plasticity within this region. So Catherine Woolley has shown in the CA3 region that spine and synapse density can vary dramatically. In the CA3 region, the late Bruce McEwen has shown that repeated chronic restraint stress can cause atrophy in the apical dendrites of males. And when I did a postdoc with them, we found that in females, the dendritic atrophy was in the basal dendrites. Same stress, but very different output. Now, some of you might be thinking, this is not so exciting. One is the apical, one's the basal dendrites. But in terms of functional impact, it actually impairs learning in males, but facilitates learning in females. And that's really interesting when the manifestation is very different of that stress. And in the dentate gyrus, this is my favorite region of the hippocampus because it maintains the ability to produce new neurons throughout the lifespan. And this is true in humans as well as in many mammalian species, despite that study coming out a couple of years ago. These neural stem cells reside in the subgranular zone. When they decide to divide, they form two daughter cells, at least one of which will become a mature granule neuron and make connections. Whether or not they're appropriate connections is an open question. And there are a number of components of neurogenesis. This is something I study. I'm not going to go through each one of these, but any change in one of these components can result in a net increase or decrease in neurogenesis. And there are a number of ways to visualize these new cells, the primary of them being something like a DNA synthesis marker like BRDU, bromodeoxyuridine. Another one is double cortin, which is an immature neuronal marker. I do use this, so you will see a couple of slides on this. The nice thing about double cortin is it's expressed for the sort of lifespan of the immature neuron. So you can see baby-like cells, as well as what I call school-age-like cells, as well as more cells that look much more like an adult, and I call them teenage cells, but they're still all immature cells. However, they will eventually become mature cells. And neurogenesis in the hippocampus has been thought of as a neural marker of depression, sort of one of those hallmarks. Why? Because this study came out, and this is Jessica Malberg from Ron Duman's lab, who showed that, this is using the SSRI Prozac or fluoxetine, that chronic exposure to this drug would increase cell proliferation, but not acute exposure. And this caused quite a splash because the behavioral alleviation of depression from antidepressants occurs at a much later time point. You can alter serotonin very quickly using SSRIs, but this neuroplasticity effect takes some weeks, which aligns with the behavioral alleviation. Exercise is thought of as a mild antidepressant. At least that's what I tell myself every time I force myself to go for a run. But it also increases neurogenesis. And things that we know contribute to depression, like chronic stress hormones, fluctuating estradiol, they all reduce neurogenesis. And the last point I want to make here is that in depressed patients, as well as in every single animal model of depression, you see a reduction in neurogenesis in the hippocampus. I think that's really important to point out because we often see a lack of translation, I'll call it, from in animal models to disease. But in this case, we have some very good evidence to support this. So you see that here's Ki67, which is a marker of cell proliferation. So in people with major depressive disorder, you see reduction in cell proliferation. Now, in this particular paper, they looked at tricyclic antidepressants versus SSRI, selective serotonin reuptake inhibitors, and found sort of an overshoot here. I wouldn't make too much of it. They don't always see this, but it stimulated a study in my own lab to look at sex differences, and I'll talk about that in just a second. When people have actually reduced neurogenesis to determine if they are necessary for depression, they don't see that. So if you reduce neurogenesis, you don't always see a depressive-like endophenotype. But what you do see is that it affects antidepressant efficacy. So if you reduce neurogenesis using and you almost eliminate it. It's reduced that much. Using X-ray radiation in both of these studies, you find that it reduces F, and so this is fluoxetine, this is imipramine, which work in normal animals, but not where neurogenesis is significantly reduced. So it's blocking these antidepressants to influence anxiety-like behavior. This also showing something very similar. So this is looking at DEX-induced cohort suppression. So you want to be able to see a lot of suppression. That indicates a proper negative feedback, which is what you see with fluoxetine in red here, coupled with unpredictable chronic stress. But in the radiated group with very low levels of neurogenesis, fluoxetine does not work. So neurogenesis seems to be linked to antidepressant efficacy. So depression and inflammation, we know from a number of meta-analyses that a number of pro-inflammatory cytokines are increased with depression, major depressive disorder, particularly IL-6, IL-1 beta. You can also look at microglia, which are the brain's resident immune cells, and you can look at the morphology of these cells. These cells also emit cytokines. That's why they're interesting to look at. And if you see more of this ramified activated state, the amoeboid state, you see that should sort of be associated with more pro-inflammatory cytokines. And this study came out a year ago, and I thought it was really interesting because in terms of antidepressant efficacy, it's the non-responders that we have there in pink that actually increase in these pro-inflammatory cytokines, not decrease, but increase, while in responders, there's either a decrease or they don't change. So suggesting that inflammation might actually be a marker of non-responding. So coming back to the question of, does sex influence the ability of antidepressants to increase neurogenesis in the dentate gyrus? We did this study a couple of years ago, now, or 7 years ago, I guess now, looking at postmortem tissue from the Stanley Medical Research Foundation. And they have 3 groups: controls, depressed patients that were prescribed antidepressants, depressed patients with psychotic symptoms that were prescribed both antidepressants and antipsychotics. And this is work of John Epp, who did his PhD in my lab, who's now at the University of Calgary. And we looked at those double cortin expressing cells, so an endogenous marker of immature neurons. And what we saw is no big difference among the men in the group. You can actually see a decrease here, but it was the women that showed this upregulation with antidepressants. And we also saw the upregulation of these immature cells in relation to antidepressants was only seen in the lower than 50 group, not in the above 50 group. Unfortunately, we didn't have enough of an N to split it up by age and sex. That would have been really nice. But individually, we know that both aging and sex influences the ability of antidepressants to increase neurogenesis in the hippocampus of patients themselves. Very recently, I have a postdoc in the lab, Travis Hodges, who's been looking at sex differences and negative cognitive bias. And I'll just show you a tiny little snippet of his work. So negative cognitive bias is the perception of ambiguous stimuli, situations, or events as negative. And it's associated with severity of depression and can predict the onset of future depressive episodes, at least in adolescent females. So Travis has been looking at both sex and age-specific neurobiology underlying negative cognitive bias in rats. And he's doing this because we want to lay the foundation for developing novel precision therapeutics for reducing negative cognitive bias. And so the way he's been doing this is using a fear-based cognitive task. So it's a lot of training. You pair one context with a shock and a very different context without a shock. And you do this over several days, 16 days to be exact. And then sometime later, you present them with an ambiguous context. So something in between the two contexts. And as I said, he's been doing it across sexes and across age. And then you can look to see their freezing behavior. To the ambiguous context, are they displaying high freezing, which would indicate a negative bias, or lower freezing, which would indicate a positive bias? And then he's been looking at neural activity using CFOS, which is an immediate early gene. And what he's found is this is a freezing on day 16 to the shock context versus a non-shock context. Not surprisingly, more freezing in the shock context, less freezing in the no-shock context. But what happens when you give an ambiguous context? Adolescents were quite positive. They had quite a positive bias. In young adults, it starts to get more negative. And in middle age, this was very surprising to me. We saw a negative bias in the males, but more of a positive bias in the females. And Travis has run a discrimination index here. So you can see this sort of age-wise progression of this negative kind of bias with age in males, but you don't see that in females. Like I said, it surprised me. I thought that females would show more, maybe because I'm getting older and I can be quite grouchy, but we didn't see that. We saw that it was the males that had more of a cognitive bias. And then I was thinking, in the Hollywood, at there is this idea of grumpy old men. So I told Travis that I thought he had a behavioral indices of grumpy old men. As I said, he's also been looking at immediate early genes, and we've been looking at functional connectivity. And so here we're just looking at how the activity patterns correlate with other regions. And he's been looking at the amygdala as well as many regions in the hippocampus. And perhaps surprisingly, he sees completely opposite functional connectivity. So the regions might be similar in terms of what's activated, but you see negative correlations in the males and positive correlations in the females. And this reminds me to tell you that there are many types of sex differences. Here we see, quantitative differences in, more females and males showing depression. You can also see population differences. So females, for example, show more anxiety coupled with depression than males do. But you can also see that underlying neural mechanisms might differ. And that's something that Travis has been looking at. Okay, so I think sex differences are really important, but it's also important to dive into single sex studies. Why are women more susceptible to depression than men? So we've created some animal models to look specifically at that. And you might think, okay, ovarian hormones are high here, so high ovarian hormones, higher depression. But in fact, the time of greatest risk in a woman's lifetime is during the postpartum period for de novo depression. And look at what happens during postpartum. They go down quite a bit. Same thing in perimenopause. You see a reduction in estradiol levels rather than an increase. So we've also, as I said, want to look at antidepressant efficacy. So we chose perinatal depression. Why? Because, as I said, it's the time of greatest risk in a woman's lifetime to develop depression. And the DSM-5 says that this has to be depression that occurs during gestation or up to 4 weeks postpartum. But in fact, depression onset during pregnancy versus the postpartum can be very different. So depression onset during pregnancy is associated with history of depression, whereas depression onset during postpartum is not so much. So there's a low risk for de novo depression during pregnancy, but a high risk for de novo depression during the postpartum. And in fact, that accounts for 40% of all perinatal depression. So here's a chart to show this. This is the risk for first-time hospital admission for any mental disorder in mothers. And what you can see in that pink outline bar is that the highest risk is in that first 3 months after depression, not during pregnancy at all. In fact, there's a much lower risk to develop depression. So we were really wanting to focus on that de novo depression. And before I describe our models, I just thought I'd quickly give you a little funny joke, but to prove a point. And that is, so this is a statue that is on the island of Malta, and they found many statues like this of pregnant women. I know you can't tell it's a pregnant woman because just the bottom half of her. I'll have to find a better example of that. And then I think we went through a dark period in our history where women were sequestered during pregnancy, weren't allowed to see them. And then Demi Moore posed on the cover of Vanity Fair in 1991 in all her pregnant glory. And I mean, this was shocking at the time in the world. And now you can't go by a checkout stand or a Hollywood show without seeing the latest celebrity that is pregnant, and they look amazing in all their pregnant glory. And so I think there's this idea that pregnancy is the most amazing thing. So I thought I'd share with you the worst picture of me ever taken in my life to prove a point, which is that it takes a massive toll on a woman's body. I know this is unfair because I've just given birth. I'm not pregnant, but I don't have any makeup on. I look awful. But the point is that there are so many changes that occur in the woman's body, and especially with hormones, you have very dramatic increases during pregnancy, and then they plummet during the postpartum period. And so it shouldn't be so surprising that there might be some repercussions in terms of disease. So in the short term, postpartum depression, psychosis, obsessive compulsive disorder is 3 times more likely in the postpartum than in any other time period. We're also very recently looking at more longer term impacts on obesity, metabolic syndrome, and brain aging. And there's some really lovely work coming out of the UK on this by Anne-Marie Delang. You also see decreased risk for a variety of cancers, and I think that's also really important to point out. In other words, motherhood can impact our health, both in the short term and long term. And what's really fascinating is that the biological outcomes that are associated with pregnancy and the postpartum are very much mirror what's happening with depression. So you see decreases in hippocampal volume, you see increases in cortisol, increases in pro-inflammatory markers, and increases in tryptophan metabolism, suggesting that serotonin levels are decreased. So maybe it shouldn't be, again, so surprising that this might be a time of great risk for women to develop depression. So I do think that these changes in neuroplasticity, these high levels, and then this withdrawal with high levels makes women more vulnerable to postpartum depression. And we know from the work of Oatridge, who showed a total brain volume decrease of about 6%. It bounces back postpartum, this time 0 is parturition. This is the work of Hoxima, who showed compared to pre-pregnancy, 2 months post-pregnancy is associated with a reduction in hippocampal volume that partially recovers 2 years later. And we see something very similar in terms of neurogenesis in the rodent. We see very low levels of new neurons in, this is primiparous rats, and it's due to pregnancy alone. So this is animals that were pregnant where we remove their pups. And so we can say that it is, we think the pregnancy hormones that's causing this reduction in neurogenesis that might be influencing volume. We also see changes in the immune system. So Rand Eade and Benedetta Lunar in her own lab in Ohio State have shown more activated microglia in the early postpartum stages. We also see serum levels of one of the pro-inflammatory cytokines that's associated with depression are increased in the early postpartum period in rats. And Paulo Duarte Guterman, who's doing a postdoc in my lab, has found in the late postpartum indices of, depending on parity, indices of higher levels of pro-inflammatory cytokines in the hippocampus. So there's lots of, not only is perinatal depression heterogeneous disease, but there's lots of biomarkers that naturally occur during the postpartum period that are also seen in depression. So because de novo depression in the postpartum accounts for 40% of all perinatal depression, that's what we wanted to focus on. And so we created 2 animal models of de novo postpartum depression, one based on hormonal withdrawal after birth, very high levels during pregnancy, and then they withdraw, and the other one based on increased corticosterone during the postpartum to model both early and late postpartum symptoms. So I'm going to quickly go through this because it's some older work that we've done, but we asked the question, does withdrawal from a hormone-stimulated pregnancy make females more vulnerable or susceptible to depressive-like endophenotype? And before I go through this, important to understand, and we don't do this in all studies, but we look at a number of behaviors. I'm happy to answer questions about this afterwards. We look at endocrine outputs. We look at hippocampus, not just hippocampus anymore. We also look at amygdala and the frontal cortex. Again, happy to answer questions about that. And we look at dorsal versus ventral hippocampus, thought to be more important for stress regulation. Inflammation, both circulating levels and in the brain. And we've just started to look at that tryptophan kynurenine pathway, which is a sort of a way to look at serotonin signaling. So withdrawal from a hormone signaling pregnancy increased a depressive-like phenotype. So we look at increased passive coping in the forced swim test, decreased sucrose preference, and it decreased plasticity in the hippocampus, most notably neurogenesis. And we could rescue this. We could rescue this either with maintaining them at this high levels of hormones, or by giving them an estrogen receptor agonist, or by giving them a tricyclic antidepressant, imipramine. And I show this because there's a lot of good human data showing the same kinds of things. So this is two papers quite a bit far apart, but what you can see is that in women with a history of depression, when they withdrew them from very high levels of ovarian hormones, and they do this by using a GnRH agonist, these women showed greater depression as measured by this scale in women with a history of postpartum depression, most notably, but also in this comparison group, you see this small increase, which is the exact same thing that Djavad Mowafaghian showed just very recently. And this is using the Hamilton Depression Score. These are women that have not had a history of depression. And you can see if you withdraw them from ovarian hormones, there's more endorsement of depressive symptoms. They're not depressed, but you can see an increase in these scores. And this GnRH agonist, this is all work by Djavad Mowafaghian, is this estradiol decrease is associated with the change in depression ratings, so depressive scores, and also with an increase in the serotonin transporter binding. So suggesting that it's associated with changes in serotonin signaling. So withdrawal from ovarian hormones increases depressive symptoms in younger women and rodents. So turning to our other model, we're looking at high levels of corticosterone in rats, but then principal glucocorticoid in humans is cortisol. And in women with depressive symptoms postpartum, you also see high levels of cortisol. So we did something very simple. We just gave high levels of cort during the postpartum period. And what we found is that it reduces maternal care, it increases passive coping in the forced swim test, and it reduces neuroplasticity. But very surprising to us, perhaps, is that fluoxetine wasn't as efficacious as we thought it would be. It was early on, so it does restore maternal care, but it doesn't restore passive coping or neuroplasticity. As I said, we've also tried exercise, and that didn't restore maternal care, but it did restore passive coping in the forced swim test and neuroplasticity. I'll just briefly show you that. So here's what we see. We've done this study quite a few times now, but with high cort during the postpartum, you see reduction in nursing in this dark bars and an increase in time spent off nest. When you give fluoxetine, which is in those hash bars, you can restore both of these deficits in maternal care. Here's for swim test. So we see an increase in passive coping with the cort in that high dark gray bar, and fluoxetine in the hash bars, you see it does nothing. It actually, if anything, increases passive coping. And exercise, though, does seem to decrease passive coping. When we look at neurogenesis, I know there's a very busy slide because it's divided into dorsal and ventral regions, but you can see that cort itself decreases neurogenesis, as you might expect, but fluoxetine in these hashed bars does not increase it at all. We did see an exercise by fluoxetine effect, just for those of you that might be interested in that. So in essence, fluoxetine and citrulline aren't efficacious, at least in the long-term postpartum. Why? And Wansoo Chu, who's doing a PhD in my lab, was asking this question. And she looked at inflammation, because you might remember that I talked about it being related to non-responders. And what she found, we looked in the frontal cortex and the hippocampus and in plasma. And what she found was that maternal fluoxetine, and it didn't matter whether they were given cort or not, and so in these hash bars, was associated with an increase in IL-1 beta levels in brain and hippocampus, and in plasma CXCL1, that's a chemokine. And just to remind you again, that Siad et al. Showed the same increase in IL-1 beta in circulating levels in non-responders. So we think that that might have something to do with it. The other thing that we looked at was this tryptophan kynurenine pathway. And so tryptophan is the main precursor to serotonin, but it can also go down this kynurenine pathway. So have this shunted away from serotonin. And one of the instigators of this is IL-1 beta. And in fact, this pathway has been very recently associated with responding and non-responders. And what Wansoo found was that tryptophan levels themselves were lower in the fluoxetine treated groups. So suggesting that both tryptophan metabolism and IL-1 beta might be related to this inefficacy. The other things we found that we haven't published yet, but hope to sometime soon is we've looked at receptor binding, serotonin receptor binding. We've looked at 1A, 2, and 4, and this is with the Befeuichager. I'm showing you the 4 here, but we see, because it's the nicest effects, I will say, but we do see reduced serotonin signaling with the paroxetine. There's some fluoxetine here, but fluoxetine. So in an interim conclusion here, maternal corticosterone increases depressive-like behavior, reduces maternal care, reduces plasticity, and antidepressants are only able to reverse maternal care in this model. And we think that is related to both inflammation effects that we see and effects on the tryptophan kynurenine pathway, and possibly also the serotonin receptor binding. So those are the sort of areas that we're looking into. And I didn't want to leave this section without any hope. There's some very recent data based on some animal models coming out of Jamie McGuire's lab showing that brexanolone, which is an analog of allopurinol, which is a metabolite of progesterone, can improve perinatal depression, mostly postpartum depression, in women. Some very nice data coming out of the US. I'm going to end off by 2 last studies. One is I want to look at SSRI efficacy outside of the postpartum. Do we see efficacy of SSRIs in females? Doesn't look like it's so efficacious in the postpartum, but what about in other time periods? And so Rand E. did this work, and she gave 6 weeks of chronic unpredictable stress to mimic depression, depressive-like endophenotypes. And in the last 3 weeks, she gave fluoxetine or an SSRI, again, to sort of mimic more what you'd see in humans. And then she looked at a variety of behaviors and neural markers. And there's a very busy slide, but it's very clear data, I'd say. So the overoptimized animals, so I had this idea that ovarian hormones would predict antidepressant efficacy. So what we found very clearly was ovarian hormones did predict susceptibility to depressive-like endophenotypes after chronic unpredictable stress. So in green there, you see the overoptimized group, and you can see more depression in the post-swim test. We saw sucrose anhedonia. In the green or OVEX group, we saw increased anxiety and the novelty suppressed feeding. And we also showed impairments in negative feedback using the dexamethasone suppression test. But remember, we're talking about antidepressant efficacy here. So what did we see? Well, again, busy slide, but SSRIs didn't afford any behavioral efficacy. So if anything, we increased passive immobility or passive coping. We increased anxiety, at least in the shams. And maybe there's a slight increase, but it wasn't significant in the overectomy group to reduce sucrose preference. But we did improve, fluoxetine did improve HPA negative feedback to a degree. And this reminds me, I thought there'd be a big difference between the 2 groups in terms of antidepressant efficacy. And, I was wrong. And I love this quote because it's the great tragedy of science is the slaying of a beautiful hypothesis by an ugly fact. And I would say that is a pretty ugly fact. In the interest of time, I'm going to not show that data. But this is just showing that in the brain, we did see some efficacy of SSRIs. So along with HPA negative feedback, we see that cell proliferation is also increased. And we saw microglia were decreased. So with longer term withdrawal from ovarian hormones, we saw an increased expression of depressive-like endophenotypes, but a limit of the efficacy of fluoxetine. Okay, so oops. So in the last 3 minutes or so, I'm going to say, because I've talked a lot about females, I think it's really important to have some look at so the reasons why females are more susceptible to depression, but why are males less susceptible to depression? And so Stephen Wainwright, who did a PhD in my lab, looked at this question. And basically, we asked the same kind of question. Instead of doing an ovariectomy, we did castration and again gave them unpredictable stress, and sometimes gave them either imipramine or testosterone to see if we could shoot this. And what he found is that testicular hormones do make males less vulnerable to stress and depressive-like endophenotype. So when they were castrated, we saw an increase in passive immobility, increase in anxiety and the novelty-suppressed feeding, and a decrease in neurogenesis. And when we gave them back either testosterone or we didn't castrate them, we saw that this was enough to alleviate these symptoms or endophenotypes of depression. It didn't always work, and sometimes imimidine worked and sometimes testosterone worked. It was a tiny bit messy, but what was very clear was that testicular hormones do interact with stress to make males less vulnerable to the negative effects of stress. And then the last study I wanted to tell you about was because I fixate on neurogenesis, but obviously there are many other forms of neuroplasticity, and one is PSA-NCAM. PSA-NCAM allows for NCAM-NCAM interactions to be plastic, to remodel. So PSA-NCAM is this green fuzzy stuff here, and so it's there, it allows the synapse to remodel. If you take it away using a bacterial endophage called ENDO-N, it makes these interactions sticky, so there's no longer any plasticity in these NCAM-NCAM interactions or synapses. And so what Steve did was something akin to what Rand did. So he gave 6 weeks of chronic unpredictable stress, and in the last 3 weeks gave treatment of fluoxetine and did a variety of behavioral tests and looked at these markers. But in half of the animals, he gave them this bacteriophage ENDO-N to get rid of that PSA-NCAM. And I wouldn't be showing you this if we didn't see some beautiful results. So we found that this is under chronic unpredictable stress. Fluoxetine, unlike what you see in females, you see an increase in sucrose preference, you see a reduction in passive coping and immobility. But if the animals had PSA-NCAM eliminated with ENDO-N, you see that fluoxetine was not allowed, did not show any behavioral efficacy either for sucrose preference or for immobility. And just to show you one measure of neuroplasticity, we looked at PSD95, which is a synaptic protein. And we saw here, you see fluoxetine increases PSD95 in different regions of the hippocampus, but with ENDO-N, it no longer increases neuroplasticity. So just, I think simply, PSA-NCAM mediates antidepressant efficacy in an animal model of depression in males, and we haven't tried it in females yet, but I'd like to do so, although we don't see a lot of antidepressant efficacy in females, at least in our hands. So in summary then, I'm sorry, I know I talk a little fast. I meant to try to slow down a little bit, but I'm happy to answer any questions. We do see that low ovarectomy or castration results in greater susceptibility to develop depressive-like endophenotypes after chronic stress. And we see that fluoxetine or SSRIs are less efficacious in females compared to males. We think this might be related to inflammation, tryptophan, and neurogenesis, at least in the postpartum, and that's something that we're following up on. And I think one of the more exciting findings is also that PSA-NCAM is related to antidepressant efficacy, at least in males. And with that, I'd like to thank the wonderful people in my lab that do the work, including Adenia Islas, who is recently a research scientist in Mexico, so has gone back to Mexico. And obviously, I want to thank the people that have given me funding throughout the years, too. I don't get funding from all of these people now. It's just based on past work. And with that, I shall conclude. Should I stop sharing? I shall stop sharing.
Thank you very much, Liisa Galea, for this interesting talk. So now is the session of questions. Okay. Questions, my dear? Don't be shy.
Alonso. Hopefully you can ask in English because my Spanish is rusty.
Yes, Liisa. Thank you very much for your talk. It is always very interesting to hear you.
Thank you.
I always learn from your talks.
Oh, good.
Are so nice. I wanted to ask, in the Travis study, if these age females were or were not ovariectomized.
They were not.
They were
right.
not.
So that's a very good question. So that is something, obviously, given our past work, we'd like to do. And you're right. It's if, based on what we found before, if we overactimize them, they should show more of a negative bias. And none of those were under any kind of model of depression. So we have done the study, and I think it was in young adults, where he has looked at chronic unpredictable stress, but it's not ready for prime time yet. So he's been concentrating on getting this particular paper out. Yeah, but yeah, in that, my quick look at that data, and I haven't spent a lot of time looking at it though, it does look like females are more affected by the chronic unpredictable stress and towards a more negative bias, but don't quote me on that yet. But no, that's a really good question. I think that's something that we need to consider. And he does have all the blood, so we can look at hormone levels and estrous cycle. So we can look at all of that, but we haven't done a good look at it yet.
Because these middle-aged rats do not lose the ability to produce estrogens, no? So
that's great. That's right. I mean,
so probably that makes the difference with males. I mean, I don't know why. If they are middle-aged animals, probably they are in constant estrus. Or probably even if they are not in constant estrus, they have quite high levels of estrogen.
Yeah, depending on whether they're in persistent diestrus or estrus. So the one that Rand did, and I didn't show that graph actually, but they were, we had shams and we had overectomy, but the sham animals still had quite low levels of estradiol. They were much lower than you'd see in a normal young adult. So it depends on whether they, what sort of phase they were in, but we can easily look at that.
May I make another question? Or?
Sure.
Yes,
well, I mean, I shouldn't say that. This is up to Sandra. I'm okay.
The student is thinking the question, so you can use the same.
Yeah.
I thought that postpartum depression was somehow predicted by premenstrual depression. I mean, that women that used to have premenstrual depression most likely would have postpartum depression and not necessarily pregnant depression. Or
that's a very good observation, and I am not the best one to answer that. I know that there are at least one person online, Dania Islas, who could answer that question better than I could. There definitely is a link. I don't know if Dania is probably too shy to speak up, but there is a link. She is writing a review right now, aren't you, Dania, about this exact topic? But I don't know that it's as well studied as it could be. Do you want to answer that, Dania?
Yeah, well,
even menopause or postmenopause depression, no? Perimenopause depression.
Oh yeah, that too. Yeah.
All these three were linked as those women were particularly susceptible to progesterone, or probably both estrogen and progesterone withdrew. No?
Yeah, Dania, you can go. Yeah, I need your wisdom. Save me.
Well, I wasn't ready, but hi, Dr. Alonso, how are you?
Hello, Dania, how are you?
Oh, good. And hello, Liisa as well, and Sandra and everyone. Okay.
Hello.
So yeah, previous studies have shown that it is actually an increase in the risk for having postpartum depression when you have PMDD or premenstrual dysphoric disorder. Why is happening is happening this? We don't know exactly. We're trying to build a story in which chronic stress probably might be triggering this susceptibility for having premenstrual anxiety or premenstrual depression, and then goes to postpartum depression. And also, which is interesting as well, is that postpartum depression can trigger also perimenopause depression. Mm-hmm. So apparently it's like a cascade, which we don't know exactly why is happening. Actually.
So PMDD, is it allowed that the speaker asks questions? PMDD, is it associated with a greater risk of depression during pregnancy, or is it more specific to postpartum?
It is more specific for postpartum.
Yeah,
yes.
That's what Alonso says. And yes. I mean, I think that makes sense, right? If you're talking about that sort of susceptibility to that withdrawal of hormones, you'd expect that to be true in all of those different time courses, which is really...
Yeah, apparently the withdrawal is making something, of course, like the high fluctuation followed by withdrawal of these hormones, the key of this thing. Not only the withdrawal, I mean, it's a combination between the exposition to high levels of hormones, like happenings in the late luteal phase, and then in the pregnancy, and then followed by a abrupt withdrawal. That would be both.
Thank you for saving me, Dania.
Okay. Well, another question?
Yes.
Otherwise, we're going to stick on estrogens, and which is one of my favorite topics. So I'm totally fine with it. It was hard for me to do the study on that Steve wanted to do on testosterone and testicular hormones. I was like, really? I like estrogens, but you know, testosterone gets converted to estradiol, so it was okay in the end of it.
May I ask a last question?
Let me, Viridiana, and
yes, of course, please.
Viridiana.
Yeah.
Hello.
Hi.
I want to know, well, related to these changes in the hormonal levels, I'm curious about the polycystic ovary syndrome. I don't know if there is any animal models to show changes in the neurogenesis.
Yeah, that's a really, you're not going to like my answer, which is I don't know, but I have seen more articles about polycystic ovarian syndrome very recently. There's a journal I edit called Frontiers in Neurotechnology, and there was one very recently by Frida Zarabji. Now, it obviously wouldn't have been probably about depression, although she's been doing some depression work actually post-stroke. She does stroke models. But I think she was talking about animal I should know all of the papers that get published there, but I don't. I think she was talking about some of those. So,
but not about neurogenesis.
I mean, I don't quote me on that. I don't know. I can, polycystic ovarian syndrome is associated with high androgens. Is that correct?
Yes, high androgens and low estrogens.
Right.
So that's why I'm curious.
Yeah, so the only thing I can tell you that we've done ourselves is, actually there's a couple of studies, one that's published, both of them are published, what am I talking about? Sometimes I forget, but it's not a model of polycystic ovarian syndrome, but we've given DHT, so that dihydrotestosterone, to females, and it doesn't do anything either in young adults or middle age in terms of neurogenesis. But we've given letrozole to middle-aged mice, female mice, and that which blocks conversion of testosterone to estradiol, right? Blocks hormones. That increased neurogenesis, surprisingly. So there could be something, but we don't know why letrozole worked, but DHT doesn't.
Thank you.
Good area to work in.
Yes, I think.
Okay, anything else? Yes. No, you can go to Canada and ask directly.
Alonso, did you want to ask questions or time or no?
Well,
yes, I would like to ask why fluoxetine?
Sorry?
Well, in one of the very nice studies, you showed that fluoxetine lacked an effect in a particular model with females treated with corticosteroid.
Yeah.
Probably because of the high levels of cytokines, no?
I think so. I mean, we don't know for sure, but yeah.
But then in another model, also fluoxetine lacked an effect in females, in 8 females.
That's true.
And why? I mean, I'm always concerned by the observation of your group that fluoxetine lacked an effect in females, where part of the world states that fluoxetine has a larger effect in females.
I know. When you look at the human literature, though, it's not super clear. But yeah, I mean, I've written that too, that, you see a greater effect. Oh, so the one slide that I said, I don't have time, and it's just that time I'm not going to show it. So that study that you're talking about, and that was in middle-aged females too. We had sham and overectomy with the fluoxetine. And I put that in there to specifically show that when you looked at a correlation between estradiol and passive coping in the forced swim test, you did see that fluoxetine worked better in the higher estradiol group. So it works with that that idea that if you have higher levels of estradiol, SSRI should work better. I can show you that slide if you want me to show you. But so if you look a little deeper, you do see those connections. It's just overall with all those groups, we didn't see a positive effect on, and it could be the dose too. I mean, that's also a possibility, right? We don't do dose response.
The fluoxetine dose, you mean?
Yeah, it could be the fluoxetine dose. Yeah.
Which one are you using?
Sorry?
Which dose are you using?
I think it's 10 milligrams per kilogram. Yeah.
It seems to be quite okay. I mean, that's not.
Yeah, it should be okay. Do we have time for me to show that slide or not, Sandra? I don't have to show that slide. It is published in the paper, so.
I can look directly.
So sometimes we do see that. The other thing that a lot in the literature, it's not always the case that people are looking long term. So we do see efficacy with fluoxetine short term on maternal care. And we don't know if that's because we see fluoxetine court reduces maternal care, but fluoxetine boosts that. So it rescues that effect. And I don't know if that's because it works quickly and, for a short period of time. And then if you lose efficacy long-term, that losing efficacy long-term, you do see in humans as well, right? So I wonder if that has something to do with it, but I don't know. The other pet theory I have that I have not done, but I do think there might be something to it, and have at it because I can't do everything, is as SSRIs do seem to lose efficacy after a while, even in humans, I think it's like a third lose efficacy after a while. I wonder what it does to sex hormone levels themselves, right? It affects sexual behavior, motivation, and if it is dampening testosterone or dampening estradiol, that might be one reason why you see that efficacy being lost.
Those effects. Okay. Thank you, Liisa. Thank you very much. Thank you, Sandra. Very nice meeting you as always.
Okay, one question for the most of the audience is students, undergraduate students. So they ask how depressive phenotype in rats, and there are another animal model for depressions or and it is I think this is an interesting question. No? Have you assessed the effect of the lactation on postpartum depression?
Yes. I see it in the chat. Yeah, those are really good questions. So I probably went over that slide really quickly, but the depressive phenotype, we try to do a lot of things because if you concentrate on only one thing, you might not see effects. Or it's not that you might not see it, but that it's it's a broad spectrum disease, right? It's not just affecting one thing. Right, to question the whether the test that you're using is good for both males and females, because it's not always the case. So for forced swim test, it seems to affect them both the same. You see this increase in immobility. I would say forced swim test is very good in rats. I don't like it so much in mice. You see very high levels in mice of immobility just normally, whereas in rats you'll see like 20% if you haven't done anything to it, whereas you see almost like 80% sometimes in mice, and it probably depends on the strain of mice. I haven't done as much work in mice, but for the novelty suppressed feeding test, which is an open field, they're food deprived, and then you put food in the middle. And the first time we did it in males was fine. We got the result we expected to find. When we first did it in females, they didn't move at all. Like they didn't care. They were hungry, they were motivated to go, but they wouldn't. We didn't know if is it too bright or is it the food. So we did a little bit more reading and we gave them Fruit Loops instead of rat chow, and then they moved. then we saw the effects that we expected to see with non-obtuse breastfeeding. So you're absolutely right. Sometimes you have to tweak the test a little bit so that it will work effectively in either sex. And are there other animal models of depression and the assessment is similar? Yeah, CORT is very good. A lot of people don't like it because it's not stress, but CORT and chronic unpredictable stress, I think, are very good models of depression. Just one thing to look out for in the literature, some people use very short paradigms for chronic unpredictable stress. Some people use longer ones. We've done both. And I didn't show you the short one because it is definitely different. Like, so Eric Nestler's done a very good job using a short paradigm. I think it's 10 days. Georgia Hodes and Russ Romeo, I think, were involved in those. And they have a short unpredictable stress, but we couldn't get the same kind of phenotype going in our with it. So I think, you must have to play around with the stresses that you use. So it really depends if you're using short or longer. And then in terms of the effect of lactation, we've done a few things. So we can look at body weight. Are they gaining as much in the CORT treated DAMs versus the not and the fluoxetine treated grams? And we do see we see less nursing for sure, nursing behaviors in the CORT treated DAMs. And we do see body weight is decreased. Sometimes it's significant, sometimes it's not, but we do see it. They catch up. Like, so once they're not just taking milk, they will eat and they catch up in their body weight. So yes, in terms of fluoxetine, I don't think we don't see any changes in nursing behaviors, and I don't think we've seen any changes in body weight either. The other study we've done was just looking at, is CORT, do you see CORT in the breast milk or not? Do you see fluoxetine? We haven't done the fluoxetine one, but we've done the CORT one. And yes, we do see it in the milk, but.
Thank you very much. The last question.
You're welcome, Diana.
Okay. Well, nobody. Okay. Well, the Autonomous University of Baja California thank you very much to Liisa Galea for the talk, Stress, Depression, and Decaying Hormones in the program of the Internalización en Casa Modalidad Virtual 2020. Here, Tijuana, Baja California, 23 November of 2020. And the director signed the present certificate, Liisa Galea, and thank you very much, Liisa.
Well, thank you for inviting me. That's a beautiful certificate, actually.
Yeah, I send you.
Thank you. I'll put it up on my wall. Thank you.
Yeah.